All right, well, welcome and thank you for joining us again today. I am Dr. Carlos Vasquez. I'm the CEO at FOSSI, and it's an honor to welcome you again to another great scientific discussion within our seminar series. At FOSSI, our focus is to understand, translate, and intervene with food allergies. And one of the key questions driving our research is why has there been such a rapid increase in food allergies over the last decades? As you remember, in September, we introduced a new seminar series called Project Safe, the impact of chemicals in our food and the environment. And this series, the seminar series, it's really focused on highlighting the signs of how the evolution of chemicals in food and our environment could have an impact on human health, and especially how it may help explain some of the rapid increase in the development of food allergies and other chronic disorders. So we hope you continue to join us on our monthly seminar series where we spotlight some of the critical signs around this important topic that not only impacts food allergies, but many other indications and disorders. Today, we are thrilled to have with us Dr. Albert Laszlo Barabasi, who is a distinguished professor from the University of Northeastern University. As a professor of network science at Northeastern University. And Dr. Barabasi also holds an appointment in the Department of Medicine at Harvard Medical School. Um, he also runs the European Research Council project at the Central European University, and he has authored numerous publications and books in his areas of expertise, which centers around using network science to tackle a wide variety of scientific questions, all the way from unveiling the structure of the brain, uncovering the inner chemical architecture of the cell, and also understanding the complexity of chemicals in food. I also learned that he's also an artist on the side, so it's a great combination. Uh, so we're really thrilled to have Dr. Barabasi with us to talk today about the universe of food chemicals. And before we start, I just want to remind our audience that we're going to be taking questions from our Q&A box at the end of the seminar. So please feel free to send your questions, and we'll try to address as many as we can. But Albert, it's an honor to have you here. Thank you for spending some time with us at FOSSI.
Thanks, Carlos. And let me start sharing my screen. And it's a pleasure to be back. Back meaning that, as you know, I have had the pleasure to be part of your annual meeting, I believe, last year. So to some degree, I will kind of tell you a little bit the same story that I mentioned with some modifications. And I'm hoping also to kind of finish this talk with a challenge that I did not have last time of kind of finding avenues to work together towards allergy with the knowledge base that we built. So I'm going to talk about the foodome and network medicine. So let me just get started and say why network medicine and what is network medicine. And network medicine is simply acknowledges the fact that the components of our of any cell are not independent entities, but they're all connected to each other. And these connections is really what kind of effectively creates life. And so the Genome Project has provided us a huge amount of knowledge, but mainly provided the components of the cell. And we need another journey that we're kind of doing right now of figuring out how the pieces are connected to each other. And if you want to kind of have a high level analogy, the Genome Project has provided us the proverbial phone book, but we need a Google map to see how to find the addresses to and how to find the journey between two different components. Now, what does network medicine do? On one hand, it maps out the interactions between proteins, metabolites, genes, all the physical interactions that really are present in the cell using mainly experimental tools, but also bioinformatic information. But then once you have that, there are a couple of concepts in network medicine that are really important to understand disease, because the question comes, what is a disease? And what I'm showing you here is what is a disease in the case of asthma. What I'm highlighting here are the genes who are involved in asthma. And one of the big central discovery of network medicine is that in this very, very complex network that exists within the cell, and what you're seeing here is really the protein interaction network of the cell, the asthma-related proteins are not randomly distributed, but they tend to aggregate in a particular network neighborhood. And that network neighborhood is really where your drug targets should be, where the disease genes should be actually located. And if you ever want to kind of find a cure for asthma or biomarkers or any type of interventions, it has to focus on that particular neighborhood. The value of identifying that network neighborhood where asthma resides within the cell is that it can focus our attention, and it has major implications for how do we design, how do we identify new drug targets, how do we do drug repurposing, that is identifying drugs who could actually help, in this case, asthma patients, and understand comorbidity between diseases, and so on and so forth. Today, my goal is not to speak so much to speak about network medicine, but rather to show you how we're going to use that to actually explore food. And the first question you would ask is, why food? Well, the reason we care about food is to some degree explained by genomics itself. So when we actually look what percentage of mortality in the US is explained by the US, by the genomics, the number is a remarkably small percentage. So here I'm showing you the top 10 reasons why we die in the US. And on the right-hand side, I'm showing the kind of the guesstimated genetic risk. That is what percentage of the occurrence of that disease can be explained by genetic modifications. And some of these numbers are even exaggerated. The bottom line is that a relatively small percentage of the disease causation can be reduced purely to what you inherited, your genes. So then the question is, what is the rest, right? And obviously the big question, every time a genomic talks ends on like a GWAS talk to try to find out what genes correspond to disease, this question comes up: how do you explain the rest of the variability, the rest of causation? And always the answer is environment. Now, environment has multiple components. Health is affected by stress, by sleeping pattern, by exercise. But the biggest gorilla in the room is obviously food, right? And I'm not going to have to convince this particular audience of how important role food really plays in our well-being, for no other reason but because really kind of through food we are actually modulating actually the whole organism on a daily basis. So when we kind of started to work on the aspect of network medicine to kind of extend these tools, we said, how do we incorporate the concept of food into our genomic and network picture? And then we said, well, that's very easy because we know what's in the food, right? So let's see what the molecules that are in the food do to our health. So first, let's go ahead and figure out what molecules we know that are in the food. And that's where we had our first surprise. And to understand where that surprise is coming from, let me tell you a simple story, a well worked out story of how food affects health, right? Because of course, food is a source of energy. You must have it, but there are other mechanisms through which food affects health. And one of them is actually well known, that people who have kind of eat too much red meat, right? Then that can often lead to heart disease. And the question is, what's the mechanism? And one mechanism is a cholesterol-based mechanism, right? Cholesterol accumulates, but the other one is the pathway that I'm showing you here that has been worked out kind of 20 years ago, which is that red meat contains cholinase and L-carnitine, that if you consume red meat, then actually your gut bacteria takes these molecules and turns them into TMA. And the TMA is processed by your liver and becomes TMAO. And individuals who have an elevated level of TMAO level are 40% higher to die from heart disease than individuals who don't have an elevated level. Yeah. So this is kind of one pathway that has been well understood, and TMAO is now actually used as a biomarker for your risk of death from heart disease. But interestingly, Mediterranean diet recommends for us to eat red meat. And how does it bypass this problem? Well, because it combines red meat with garlic and olive oil, and the active ingredient of garlic is allicin, and you have the DMB actually in olive oil. And what these two molecules do is that they block the gut bacteria's ability to create TMA. So even though you consume choline and carnitine, you don't get TMA and you don't get elevated level of TMA out of it if you combine it with garlic and olive oil. So this is a well-studied pathway, and we said, okay, then let's see how much of these molecules we have in our diets. What food contains these molecules other than obviously red meat? And that was the first surprise when we realized that of the 6 chemicals involved in this pathway, only one, choline, are tracked in nutritional databases. And what is a nutritional database? Well, the FDA actually, and the USDA, I'm sorry, kind of tracks the nutritional component of all the foods in the US. But of these 5 molecules, 6 molecules, only one of them is tracked. The rest is what we call undetected. And then we said, well, how is that and why is that? And that came the second surprise because we realized that really nutrition focuses on 150 nutrients. These are typically the chemicals that you must have in your diet for you to survive. These are typically the energy sources, the fats and sugars, right, that you need on a daily basis. There are also proteins and amino acids, but they also include the vitamins. They include all the molecules that if you lack in your diet, you will not be healthy, right? And so, for example, when you look at garlic, it has 67 nutrients. But then we started to explore the other side, is to say, when people study food chemically, how many chemicals they find. And then we realized that there are an exceptional number of chemicals that are not called nutritional component, yet they are part of our food. When we started this journey, and then we published this paper actually in the very first issue of Nature Food, we were able to identify 26,000 chemicals that were shown to be present in one or some other food. Today, that started the journey through which we built a database that today contains more than 135,000 chemicals. So, for example, in our database, garlic not only has the 67 nutrients, but has 5,600 additional compounds. Now, why didn't nutrition focus on these? Well, because individually, none of these, if it's missing from your diet, will necessarily kill you or cause a disease. But it doesn't mean that these chemicals are not related to health. And I will show you evidence how many of these molecules are actually health related. And we think that more than half have some health consequences. How does this look like? Let me show you an example. Sweet basil actually has in our database about 2,100 chemical compounds. That includes flavor components. These are the volatile components that are responsible for the flavor of the basil. Then we have several of them actually, like 381, who have a disease connections, that is chemicals that are not nutritional components. Yet we know that if you consume those chemicals, that has some implications for some disease or some therapy, or there are some other therapeutic links, and also could have other health implications. So so now, what kind of classes of chemicals we have? We just have a paper very soon. It was just accepted last week in New England Journal of Medicine, where we kind of classify this, and this is mostly for the chemist in the room, of what classes of chemicals are. So here you are seeing the 150,000 molecules kind of clustered together using AI based on their chemical similarity. And on the right-hand side, I'm showing you for each the class of chemicals, what kind of chemicals are we talking about it. All I'm trying to show here is that there is an exceptional diversity of the chemicals that is in your food, and every meal that you eat bombards your cells with about 50,000 different chemicals. And now the good news is that we started to understand the chemical diversity of the food. We call it the dark matter of nutrition because there's lots of things we don't know about them. We know what the chemical are. We have their three-dimensional structure, but we don't know which chemical is in which food. And what do I mean by that? So for example, if you take apple, then we have certain components that are known because they were detected using mass spec and other things. But we know there are many other chemicals in the apple that are momentarily unknown for us, partly because no one looked for it. And so the question is, how can we ever complete this periodic table? And this is a journey that we've been on it for a while. And I think we believe now that the combination of mass spec tools and AI will be able to complete it. And we have not been successful of convincing NIH that this periodic table of food is really something that they need to invest on. But eventually, we humans have to achieve that if we want to understand how food affects health. And how would you do that? Well, on one end, we have the genome of everything that we eat. So we can actually look at the proteins and the metabolic pathways to infer what chemicals should be there. And then we can use mass spec to test if it's there. But most important, so some of them can be filled in from the genome, but most important, we have chemical information for about 3,000 different food items, but for each of them is incomplete. And we are convinced that using this incomplete data and using the similarity, the genomic similarity between the different food items, because they're not independent of each other, we will be able to use machine learning and AI to complete the table. And if we succeed with that, we can validate that using mass spec, and this will become really the genome of the food. And here I'm talking about, more poetically, the genome of the food, because really this is the elementary information that we should have about the food. And it's a shame that we don't, even though we have the tools to get to that. But let's assume that we got to that. How would we use this information? Like, why is this important? And one of the reasons it's important is because once you eat these chemicals, they bind to the proteins in your cell. They bind to the components of this network, and they influence the many chemical processes that take place in our health and eventually modulate our health. And let me show you how that happens through to an important example, which is the case of polyphenols. We all know that polyphenols are good for us, and this is the main reason that we are paying us to actually eat more plants and vegetables is because we should be exposed to more polyphenols. Polyphenols are this rich family of chemicals, about 8,000 compounds, that are typically generated by the secondary metabolism of plants, because as you know, plants have a much richer metabolism than mammalians, mainly because they cannot run away from predators. So all their defense mechanism is based on color, shape, taste, structure. And that therefore they need an exceptional chemical machinery to produce the color, the taste, the texture, the shape that either kind of furthers their survival as well as defends them from predators. And the question is, how do these chemicals affect our health? We always think that food is about energy. Interestingly, polyphenols are not processed by the human metabolism, or only marginally so. They are not source of energy. They're not part of the chemistry that we're normally familiar with in the metabolism. Rather, what they do is that they get through the liver, they get into the bloodstream, and eventually effectively bind to human proteins and modulate their ability to do things. So this effectively they have a regulatory effect, not a metabolic effect on the cellular activity. So how do we understand what polyphenol affects one's health? Like one way to do that is to use the tools of network medicine. So here I'm showing you the schematic version of the network that I showed you earlier, where proteins are interacting with each other. And let's take the example of green tea. The active ingredient of green tea is EGCG, right? When EGCG is known to be able to bind to 52 human proteins. So let's say that this is where the polyphenol targets of EGCG are. Then using the tools of network medicine, we look what are the disease modules that I showed you earlier that overlap with the targets of EGCG. So in this case, we will predict that disease A should be influenced by you drinking green tea, but disease B, or the green and the yellow, should not be influenced because the targets are not modulating that particular disease modules. So that's a conceptual way to do that using the tools of network medicine. Specifically, if you look at the details, EGCG targets turns out to be in the vicinity of the type 2 diabetes. And we are predicting that type 2 diabetes should be perturbed by that. And this is actually a well-known effect that people who have type 2 diabetes are recommended to actually eat green tea because it's beneficial to them. But using the same tools, we can predict what other diseases would be affected by drinking green tea. And this is the list that we actually came up with. This is purely predicted using the tools of network medicine. And what the good news is that the top 10 on this list are well-known diseases or phenotypes that are affected by drinking green tea. And under these are actually new predictions that need to be still tested. So this is kind of showing that we can use the information about the food molecule and put it into the network medicine pipeline to tell you what diseases will affect them. But not only just tells me that diseases will be affected, it will tell us how. That this gives us very precise, experimentally testable kind of hypothesis. So, and that could be actually falsified. So here's an example where we looked at how different diseases are affected by several polyphenols. And in particular, we focused on rosmarinic acid, and we predicted using the network medicine tool set that rosmarinic acid should actually affect vascular disease through platelet formation. So it's precisely what the mechanism is. And we also predicted what genes should be changing if you add rosmarinic acid to the cell line. And my colleague Joel Skazel tested that, and everything was exactly how we predicted. The changes in the network and the platelet formation were as the theory actually predicted. Why am I showing this? Because I'm showing you that there is a way for us to go from this cosmic picture of how the cell is interconnected, put in as perturbant the food molecules and tell you precisely what molecular processes in the cell affecting what disease should be affected by each of those food molecules. So this is a journey that we are on, and I'm going to tell you a little bit more towards the very end about that. But as I'm getting to the end, I haven't talked about how much nutrient is in each food, but this is something that we explored again in the Nature Food paper. The trick is what you need to know is that first of all, there is a 9 order of magnitude difference in the concentration of different chemicals. Vitamin E has 10 to the minus 7 concentration, and water has 10 to the second in a 100-gram food. But this concentration is rather fixed, meaning that the variability from one food to the other one is relatively minor and follows what we call a log normal distribution. Why is that important? Because now the American population doesn't consume any longer raw food. They consume ultra-processed food. 70% of the American calories are coming from ultra-processed food, which changes the chemical composition of the food. It moves these points around in the space, often many orders of magnitude. So, for example, using these ideas, we built a tool, an AI-based tool that can tell you how processed is any food that you have on the shelf in the because even though we know ultra processed food is bad for us, we don't have a measure of how ultra processed something is, and it's never labeled in the supermarket. So using these tools, actually just this month came out our paper in Nature Food, where we looked at the whole US supermarkets content, and we showed that in every food category you have very ultra processed and not so ultra processed versions of the same food. So that is that, you may not be aware of it and something says organic, but it could be hugely processed, right? Versus something doesn't say organic, may not be processed at all. And to even give this information back to the users, we released this website that now is getting more than 1 million hits per day, where it will tell you in your grocery store what is ultra processed and what is not, and will tell you in the same food category what would be the choice you could make to have a less ultra processed version of the same food that you would like to buy. So it's pretty amazing how many people are coming these days to that. So to kind of finish, and I know I'm slowly end of my time, what I would like to challenge this community, because you guys are bringing lots of knowledge in this space, is to think together with us how to activate this exceptional knowledge that my lab has collected on the nutritional dark matter. What is that knowledge? We have a database of 139,000 chemicals that are known to be in some food that are connected to about 3,000 common foods and about 18,000 species, right, where the food is coming from. And we are in the process of activating this knowledge to really do new projects with that. We are working with two teams to actually see which of these food molecules could serve as antibiotics. And we also have another big collaboration to predict which of these food molecules affect aging pathways and may be able to modulate longevity. But I have two dream projects that we have not started with, yet. We know that many food molecules are immune modulators, right? For curcumin to resveratrol and so on, there's already lots of evidence that food actually affects our immune system. But no, what we have now 139,000 chemicals in the food. How do we screen, either computationally, experimentally, the nutritional dark matter to identify which of these chemicals really affect our immune system? And the same is for allergens. We know that many allergens are proteins, but not only proteins are actually allergens. Many small molecules are known to actually have an allergenic effect, and I'm listing some of them. And we have here an opportunity to test all the chemicals in the food for their allergy effect, either computationally or experimentally, if the tools exist in your community to do so. This is not my area, so we are very open to collaborations with your community to see if it's feasible to test either the full set or a subset of these chemicals that we can rationally choose to see whether they affect the immune system or allergens or any other aspects of our health. So I'm going to finish actually with my collaborators and actually the statue of the vitamin C, which is, as you know, one of the vitamins in our food, as well as an immune modulator. So, and it's also a statue that I designed for a space in where Albert Szent-Györgyi, who discovered it, actually lived. Thank you very much for attention. Happy to take any questions.
Albert, fantastic. Thank you so much for an amazing presentation. Every time you talk about this, I'm always so impressed with especially the unknowns, because there's so many unknowns in the food space. Before I probably just have time for one question, but just as a reminder, everyone asks us, how can they help donate, host an event, follow us on social media, be active with us in social media, and join our webinars. Our next webinar, don't miss our next webinar, would be fantastic. Dr. Andrew Wan from Yale University on February 11th will be talking more about this and how it impacts food allergies. So please join us in that meeting. Maybe quickly, just I have many questions. One of them, I'll take one questions from our audience, which is, how do you incorporate this type of research? How do you incorporate the biotransformations of the human microbiome in this type of research, as we continue to learn every day about the microbiome and the fact that you generate so many secondary metabolites?
Yeah, very, very good question. Because indeed, the first time food meets us is really through the microbiome, right? That's the greeting committee. And many of the chemicals in the food are being broken up by the bacteria. And the answer is we don't know how that happens. And for that reason, we just started a collaboration with two groups and the company who are microbiome experts. And what we're trying to do is to select chemicals from each class of chemicals that I showed you on this map and then they will actually test experimentally whether how they affect individually the microbiome, both in kind of individual bacteria as well as in a combination of that mimics the human microbiome. So it's a fabulous question. We think a lot about it. The knowledge base is not there. It's a very open question that we would love to continue working on.
Thank you, Albert. I'll just close with one thought connecting back to your last slide, because unfortunately we don't have more time for questions, but um, this is beautiful, elegant, and impactful work that you've been doing. And as you made a point on, all these molecules can interact with proteins in our body. And as we think about food allergies, it is always interesting to us of not only how these molecules interact with our body, but how these molecules interact with each other, sometimes with proteins
Yes.
within their own system, to now serve as transport or provide a different bioavailability that now creates different interactions. And one of the key things at FOSSI is connecting how these chemicals interact on their own or interact in connection to impact the nervous and immune system that is highly connected, and we're learning every day so much in food allergies. So I really appreciate the challenge that you put at the end, Albert, because I think there's 100 of questions that we would love to continue to explore together, and really appreciate you taking the time. Thank you to our audience for joining again. I know these are short discussions, but I'm sure you would see a lot more discussions between Albert and Fossey as we try to tackle all these questions together. So, Albert, thank you so much. Uh,
Thank
Thank you to everyone for joining. Have a good one.